And
this is 9-2.
I might cover 9.3 and 9.4 today.
No. I thought you wanted people to do the Thanksgiving. Oh yeah, you need people to do the quiz thing on the board.
Yeah, that's what I was going to try to do tomorrow. These are short sections. You wouldn't be responsible for reading both tonight. You could read one tonight.
This is a 9-2.
What?
This is a 9-2.
Yeah, I'm just going back to this because it carries on into 9.3. So once again, these EGFRs, the epidermal growth factor receptor proteins, receive the epidermal growth factor. When they each receive it, they come together. That's the dimerization that you might have read about if you did your reading. Probably did not. Anyway, they come together, and they're the bottom of these proteins down here. That's a membrane. Can y'all tell that's a membrane? What type of organic molecule is the membrane made of? Lipids. Yeah, lipids. And these are proteins. So anyway, we receive the EGF, the epidermal growth factor. That's like growth hormone. It's received. These things come together. The phosphate groups are placed on the bottom of the protein here. It becomes phosphorylated. Here's a group of 3 proteins. These are called G proteins. Do you know why it's called G protein? Do you know why it's called G protein?
No.
Because it's a GTP, guanosine triphosphate, will come in there and react with it. The GTP actually replaces a GDP. So you receive the signal here, the GTP replaces the GDP. Now it's an activated G protein, this RAS protein. Another protein called RAF will come up, attached to it. This RAF is a kinase. Do you know what a kinase does?
It catalyzes something.
Kinases add phosphates to something. What do you think the RAF adds a phosphate to? MEC. So MEC, you can see it comes through the RAF and now it's got a phosphate. Now the MEC will phosphorylate something. It phosphorylates ERK. You see now the ERK's got a phosphate. And that ERK can do things. That's what the next section's about, what the ERK can do. You want to see what the ERK can do?
Who doesn't?
Okay, so here's the ERK. Remember I was talking about the ERK? So the ERK can phosphorylate this MNK1 protein. See, it's phosphorylated. The MNK1 protein can then phosphorylate this EIF4E protein, and so forth. So it's kind of like a phosphorylation cascade. A cascade is when one thing happens and another thing happens and another thing happens. So we phosphorylate the ERK phosphorylates the MINC phosphorylates the EIF, and so forth. And what is the purpose of all this? Well, it kind of changes the shape of these proteins when you phosphorylate them, and that can activate protein synthesis in the cytoplasm. So, do y'all know what mRNA is? Messenger RNA. We're going to talk about it in several chapters from now. I've got an RNA, magnetic RNA model here. This is DNA. This is RNA. The DNA can be copied into an RNA strand. DNA is double-stranded. RNA is single-stranded. This is in the nucleus of the cell. The DNA is copied into RNA. We call it mRNA, which leaves the nucleus and goes into the cytoplasm so a protein can be made. So here's the mRNA that's in the cytoplasm. It's got all these proteins stuck to it. These proteins are inhibiting it from being from going through the ribosome and making a protein. Are you with me? You seem like you're staring right here. So when this ERK starts this phosphorylation cascade, it changes the shape of these things enough where, notice the mRNA in this picture is all kind of coiled up here, but it flattens it out once we get these things phosphorylated. And now this mRNA, which was kind of in that top picture, it was kind of folded up like this. Now it's flattened out in this bottom picture and can go get made into a protein. So that's one of the functions of the ERK. It can cause protein synthesis. It can cause proteins to be made. This is so exciting. I can show the video and I can turn the light off.
Does it have to be flat for a protein to be made?
Yes, it does. It can't be made into a protein if it's coiled up like this.
Dysregulated MAPK signaling has been implicated in a wide range of cancers and occurs via multiple mechanisms, including activation of receptors and activating KRAS and BRAF mutations. These may lead to increased or uncontrolled cell proliferation, resistance to apoptosis, programmed cell death, and resistance to chemotherapy, radiotherapy, and targeted therapies. A vast array of growth factors exert their effects via the recruitment and activation of signaling molecules of this pathway, which include RAS, RAF, MEK, and ERK, also known as MAPK. MAPK signaling is activated through binding of a growth factor to the extracellular domain of the tyrosine kinase receptor. Signaling molecules GRB2 and SOS are next recruited to the internal docking site, resulting in RAS activation of the membrane. The efficiency and duration of signal transmission is regulated by the scaffolding protein kinase suppressor of RAS, KSR. RAS triggers a phosphorylation cascade involving RAF, MEK, and ERK proteins, leading to ERK activation and translocation to the nucleus. Once in the nucleus, ERK activates several transcription factors that mediate gene expression. Target genes thus activated by MAPK signaling contribute to high proliferation and survival. Genentech Bioncology is researching.
Now, that actually showed it going into the nucleus. So that was not perfect. That was not the perfect one. It showed that ERK going into the nucleus. This, what's in this picture, is actually outside of the nucleus. What was that, Thomas?
No.
Yeah, this happens outside the nucleus. So back to the slide before. See where it says ERK stimulates translation? That's what that thing was with the RNA, getting it to unfold so it can go through a ribosome. That was this part here. In the video, it showed it going into the nucleus and stimulating the DNA in the nucleus to cause cell multiplication. See, when stuff goes wrong in this pathway, let's say we made too much of this ERK, it might go in the nucleus too much and cause too much cell division. That's cancer. Cells multiplying out of control. Greta, are you with me? You seem as if you're not. So a lot of this stuff has been learned through cancer research. Now this is the one I'm working on the set of magnets for. Have you ever heard of epinephrine?
Yes.
Epinephrine is also known as adrenaline. And adrenaline, here's the way adrenaline works. When you release adrenaline, it causes your liver cells to release the sugar that they've stored. How does this happen? Well, the epinephrine binds to this epinephrine receptor on one of your liver cells. Epinephrine is a hormone called adrenaline. It's released by your adrenal glands when you're scared.
Is that what's an EpiPen?
Yes, an EpiPen has epinephrine. And so if they stick you with that EpiPen, this epinephrine will bind to a surface receptor on one of your liver cells. And it activates a G protein on the inside, which they're not showing here. And the G protein will slide along and hit this other protein called adenylyl cyclase. And that adenylyl cyclase starts breaking up ATP and changes it to AMP. How does it do that? It takes off a phosphate, it takes off another phosphate, and you're left with AMP, also known as cyclic AMP. Now, the cyclic AMP can activate a protein called glycogen phosphorylase. And when that thing becomes active, it can start tearing apart glycogen, which is stored sugar in the liver. And the glycogen becomes glucose. This is a polysaccharide. Y'all remember polysaccharides? Glycogen was one of the polysaccharides we learned about. And this polysaccharide will be torn up by this enzyme, glycogen phosphorylase, and it'll just, one at a time, tear these glucoses off from the glycogen, and those will be released. And those glucose molecules can exit the cell, and now you got sugar in your blood. And that all happens really fast. So if you're ever scared, you know, die!
I say that every single time.
If you're ever scared, your liver will squirt out a bunch of sugar into your blood.
Why?
And then you can use that sugar for energy. Your muscles can, you know, fight or flight response. And your blood becomes full of sugar, and that goes to your brain. It gets your brain working faster. You're awake. You're more awake than you were before, right, Cammie?
So it's better if you're constantly scared.
Well, the reason why we're not always like that is because it takes a toll on your cells to be in this high state. For instance, when there's a lot of sugar in your blood pressure is high. Well, high blood pressure will kill you earlier. Because it breaks your vessels, your kidneys, it'll destroy your organs, you know. So it's nice in the short term to have high blood pressure, but in the long term it's bad for you. So there's a balance there. Want to see video footage?
Heck yeah.
Oh. So hopefully I'll have these magnets by tomorrow for you.
Epinephrine is one of many hormones that is water-soluble, hydrophilic, and therefore unable to cross the hydrophobic plasma membranes of its target cells. Instead, it binds to receptor proteins located in the plasma membrane and does not enter the cell. When epinephrine binds to beta-adrenergic receptors on the liver cell, G proteins on the inner side of the cell membrane are activated. Each G protein is composed of 3 subunits, and the binding of epinephrine to its receptor protein causes one of the G protein subunits to dissociate from the other 2. The G protein subunit which dissociates from the others carries a GDP, which is replaced by GTP when the subunit is activated. The activated G protein subunit then diffuses within the plasma membrane until it encounters adenylyl cyclase, a membrane enzyme that is inactive until it interacts with the G protein subunit. When activated by the G protein subunit, adenylyl cyclase catalyzes the formation of CAMP from ATP. The CAMP formed at the inner surface of the membrane diffuses within the cytoplasm, where it binds to and activates protein kinase A, an enzyme that adds phosphate groups to specific cellular proteins. In liver cells, protein kinase A phosphorylates and thereby activates another enzyme called phosphorylase, which converts glycogen into glucose 6-phosphate. The
Wait a sec. Say it again. All right, I'll give you a call back. Okay, thanks. Sorry about that. Hold on, let me back this up.
A membrane enzyme that is inactive until it interacts with the G protein subunit. When activated by the G protein subunit, adenylyl cyclase catalyzes the formation of cAMP from ATP. The cAMP formed at the inner surface of the membrane diffuses within the cytoplasm, where it binds to and activates protein kinase A, an enzyme that adds phosphate groups to specific cellular proteins. In liver cells, protein kinase A phosphorylates and thereby activates another enzyme called phosphorylase, which converts glycogen into glucose 6-phosphate. The glucose 6-phosphate is then converted to glucose. Through this multi-step mechanism, epinephrine causes the liver to secrete glucose into the blood during the fight or flight reaction.
Is the glucose going out? That's how adrenaline works.
Where does it go if you don't have adrenaline going?
Where does it go?
Yeah. It's still going to the blood if not as quick?
The glucose?
Yeah.
It stays stored in the liver. Now, it can't. There are other things that can cause it to be released. Insulin is another protein. Have you ever heard of insulin? Insulin causes it to be stored in the liver cells. Glucagon causes it to be released. So there are other hormones other than epinephrine that can cause the glycogen to be released. Now, the signaling cascades that we're talking about, let me back this up, where we receive a signal and then all this stuff happens and there's some response. There's just thousands of different effects that can have. This is a foot of a mouse embryo, 15-day-old mouse embryo. And one of the effects of this signaling cascade is called apoptosis. Have you ever heard that word before?
Yes.
Apoptosis is cell suicide. And what will happen is all these cells that are in between the toes right here, they will kill themselves. And so the foot forms like this, but all these cells between the toes end up killing themselves, and so you have separation between your toes. This actually doesn't only happen in mouse embryos, it happens in human embryos. So a lot of your cells were born when you're an embryo, and they ended up killing themselves to make your final form. And the killing, the apoptosis that happens is directed by these signaling pathways. So these cells right here receive a signal to kill themselves, and then they disappear. If they didn't do that, you'd have webbed hands and feet.
I think that's pretty cool.
You'd be a heck of a swimmer.
Why don't we have them? They're useful.
Because they're not useful for us.
Bryce does.
We might would have all
Bryce is
had them.
keeping
I want web
They
hands.
didn't film themselves.
Apoptosis is an important cellular process that allows cells to die in a programmed fashion. Apoptosis plays an essential role in growth and development, such as in the womb, where the fetal hand starts out as web, and fingers are formed through the programmed death of the cells in web stasis. Apoptosis also plays an important role in removing faulty cells. For example, pathological apoptosis may be induced if cellular DNA is damaged beyond repair. In apoptosis, the cell is broken down from within by proteins called cascades. For apoptosis to occur, these cascades first need to be activated.
Cascade activation can happen via two distinct pathways, called the extrinsic and intrinsic pathways. The first pathway is referred to as the extrinsic pathway, because the initial signal comes from outside the cell. This pathway is often initiated by other cells, commonly by subsets of T lymphocytes.
That's a white blood cell.
These lymphocytes have a surface molecule known as Fas ligand, or Fas-L for short. The extrinsic pathway is initiated when Fas-L binds to Fas receptors on the surface of the targeted cell. This sets off a chain of intracellular events that will ultimately result in apoptosis. The sequence is mediated by a Fas-associated death domain, or FAD. In the final step of the extrinsic pathway, Cascades activate each other in a self-amplifying process called the Caspase Cascade. Apoptosis is then initiated as the active caspases begin the breakdown of cellular materials. The Caspase Cascade acts as a common endpoint for the second apoptotic pathway, known as the intrinsic pathway. As its name suggests, the intrinsic pathway is initiated by signals from within the cell. This intrinsic pathway is regulated by maintaining a balance between two sets of proteins in the mitochondrial membrane: anti-apoptotic proteins, such as BCL2 and BCLX, and pro-apoptotic proteins, such as FAX and BAC. In a healthy cell, the anti-apoptotic proteins bind to the pro-apoptotic ones, thereby blocking their action. But if a cell is damaged, or if it stops receiving survival signals, BCL2 and BCLX are blocked in turn. FAX and BAC are then free to punch a series of channels in mitochondria, allowing mitochondrial substances, such as cytochrome C, to leak out into the cytoplasm. The leaked cytochrome C binds to APAP1 proteins to create a compound that then activates the Caspase Cascade. Apoptosis plays a key role in growth, immune surveillance, and neoplastic development. In all of these processes, there can be abnormalities like too much or too little.
Alright. After that. Pretty cool though. Cells can kill themselves.
Nice.
Last thing, let's talk about autoinducers. I know this is the next section, but that's okay. Autoinducers are chemicals that cause cells to be able to sense. Here's a chemical called an autoinducer. And here's a cell, a bacteria. Bacteria like to do this. And a bacteria can produce an autoinducer protein that will leak out of the cell, and it could actually get back in this cell and cause this cell to react to it. Y'all remember autoinducers? I actually talked about them. Autocrine signaling. Y'all remember that? Now, if the cells are low in number, not much is going to happen. Most of these autoinducers are just going to float away and not much happens. But if the cells are at high density and one cell is releasing autoinducers, it'll get another cell. That cell will capture it, and it causes the cell to make more autoinducers. So it's what we call a positive feedback loop. The more autoinducers that are made and received, the more that are made. And more will be received, and then more will be made, and very quickly the number of these chemicals goes way up. And the cells can sense that. And having a high number of these autoinducers turns on specific genes that need to be active when there's a large number of cells. And so it's a way for cells to turn on specific genes when they're in high population and keep the genes off when they're in low population. So, without thinking, these cells can react to one another and know when their populations are high. Bacteria don't have to think. They don't have brains. Everything is just automatic with them.
A biofilm is a thin layer of microbial slime and proteins adhering to a surface. A biofilm forms when bacterial cells adhere to a surface and produce a matrix of extracellular polymeric substances, EPS, a sticky type of glue-like material. The bacteria remain embedded within the EPS which protects them. Biofilms can be found everywhere in the environment. They are present on teeth as dental plaque, in water supplies, air conditioning units, food processing, and many surfaces within healthcare. Inside biofilms, bacteria can communicate, live, feed, and grow. Bacteria are typically 200 times harder to kill with antibiotics or disinfectants inside a biofilm. And whilst they're alive, they remain a threat to patients. The bacteria will continue to grow until the biofilm is disturbed or free bacteria are released, transferring onto other surfaces via hands, gloves, cleaning cloths, or other materials. Whiteley Corporation is a world leader in combating biofilms and removing infectious organisms from within our hospitals and...
So, that wasn't a video though. It wasn't an ad. Lots of bacteria living together form a biofilm. That's what plaque is on your teeth. It's a whole bunch of bacteria living together, communicating with one another, making important enzymes that are needed when they're in high numbers. Okay, so I didn't quite finish. I'll finish this tomorrow, and then we'll do some magnet stuff, and you can finish the photosynthesis if you haven't done that yet. Out.